It's a real pleasure for me to have the opportunity to introduce our next speaker, Lee Hood, who, like our other speakers, has an extraordinary array of decorations and awards. I won't go try to do the list except to say that that it doesn't get better than Lee. And those of you that are teacher scholars have all made important contributions, which is to science, which is one of the reasons that you're here. Lee has obviously made extraordinary contributions, but I think it's probably fair to say that his vision and his drive and willingness to develop all kinds of technological advances as well as scientific ones have changed the world of science and biology in ways that it's hard to imagine. And for someone that's seen it from its inception all the way to now being able to really look at the human genome for individuals at a reasonable price and in a very short time, the possibilities have really become extraordinary. And Lee has not only driven that technology, but also its applications. And I think today we're going to hear about human health care and the advantages and changes that we're likely to see. Lee?
Well, thank you, John. What I'd like to do is give you my vision of how healthcare is going to change within the next 10 years or so, and do it from a personal point of view and illustrate a lot of the career choices as I made in developing the kind of opportunity we're going to talk about today. I remember when I was an assistant professor at Caltech in the early 1970s, right next to Max Delbruck, talking with him about the complications, the complexity of biology and the complexity of disease and how we were going to solve those problems. And what was really clear at that point in time is we had neither the technologies nor the strategies for dealing with that enormous biological complexity. And in fact, in '73, I remember reading Thomas Kuhn's The Structure of Scientific Revolution, which was about paradigm changes. And it made the point that paradigm changes are enormously difficult to achieve because of the basic conservatism of most scientists. And you have to overprove the point before you can really make changes. And what's been really delightful in looking back over my career is I've gotten involved with a series of 5 paradigm changes, and each of them has pointed toward beginning to solve some of the aspects of this problem of complexity. And in retrospect, the first 4 paradigm changes actually enabled the 5th one, which is this fundamental change in healthcare. So the first one that got started at Caltech, again in the early 70s, was really bringing engineering to biology. So we developed a series of instruments for DNA and protein sequencing and synthesis, and later developed the inkjet technology for large-scale DNA synthesis on arrays, and finally the nanostring technology for doing one of the first instruments for doing single-cell nucleic acid analysis and so forth. And what this instrumentation did was to, one, open up high-throughput biology, and that really enabled big data in biology. And as you'll see, that really is an important point. One of the instruments that we developed was the automated DNA sequencer, which enabled the Human Genome Project and got me involved the very beginning, 1985, in this project. And it was obvious to me that it was really a key project because it would give us the parts list that was utterly essential for doing this thing called global and holistic systems biology I'll talk a little bit later about. Again, it took 5 years to persuade a really skeptical biological community, not the least of which was NIH. They didn't fold until the very end. But, of course, the Genome Project did just what I predicted. What was interesting in creating the automated DNA sequencer is we had to put together a really good chemist, a terrific engineer, a computer scientist, and myself, a biologist. And I realized how essential in the future it would be for biology to be able to integrate all those skills to develop their technologies and strategies. And I, at that time, tried to propose to Caltech that I start a department of, I called it molecular biotechnology, and the biologists categorically vetoed that idea for reasons that I think are obvious to most of you. But Bill Gates made it possible to go to Seattle, the University of Washington, and set up there the first department ever. And this department, in the 8 years of its existence, was spectacular. It developed the first 2 key techniques for proteomics. It developed the software that fueled the entire Genome Project with regard to validation and assembly of DNA. We developed the inkjet technology printer there, and so forth. But what was really clear is the dean that hired me, talking about serendipity, agreed after 4 years he'd give me an additional floor and I could build on top of cross-disciplinary biology a systems biology department. And of course, he died in an avalanche in the Himalayas, and the next dean that came in had a totally different agenda. And, you know, it was really fortunate he had a different agenda, because what was clear in pushing systems biology was nothing like this had ever been done really before in academia. And the bureaucracy of academia was against it in virtually every way. I'll just say, you know, bureaucracies honed by the past just barely can deal with the present. Almost never can they deal with the future. So I resigned then and started a nonprofit research institute, the first Institute of Systems Biology, and developed system science there, and began in 2001 to really apply it to biology. And that moved us, in fact, to create these new disciplines of systems medicine and of something I'll call P4 medicine, and I'll explain it in a little bit more detail in just a few minutes. So systems medicine has 2 big essences. One is the big data essence, that is, every patient is going to have a virtual cloud of billions of data points. Data will be very heterogeneous, from molecular and cellular all the way up to social networks, and we'll have the wherewithal to integrate these data so that we can optimize wellness and minimize disease for the individual patient, the individual. And I'll show you that in spades at the end. Let me just say there are 2 challenges with this picture. One is how you integrate heterogeneous data, and that is one of the grand challenges of systems medicine. And the second is, in any big data set, there are enormous noise to signal problems, and how you extract very small signals from enormous noise really requires systems principles in general to make that distinction. It's why blood diagnostics, until very recently, have really failed. Now, one way to reduce the dimensionality of this, of course, is to abstract from this information what I call the network of networks. So networks are entities in living organisms that handle and process information. And they can operate at the molecular, at the chromosomal, at the cellular, at the organ, at the organism levels. And in a sense, they're seamlessly integrated to be able to carry out biological functions, both normal and in disease, they become disease perturbed. And if you can capture the nature of these perturbations in disease, you can begin to understand the earliest mechanisms of disease And equally important, you can use these for early diagnostics and the identification of drugs, therapeutic approaches to the disease. So this systems medicine, together with big data and its analytics, and together with socially activated patient networks, were three convergences that led to a medicine that was predictive, preventive, personalized, and participatory. And the really important point I would make is the patient or consumer-activated social networks are incredibly important for this new medicine because, one, they are the means by which patients can learn about the new medicine. Two, patients can do crowdsourcing and learn how to use the medicine more effectively. But three, in the future, they're going to become the advocates that are going to drive a very conservative system to change. And I'll talk a little bit more about that in just a moment. And to show you that P4 medicine differs from contemporary medicine in significant ways, one, it's proactive, not reactive. Two, it's focused on the individual. Three, it's focused on wellness and not just on disease. And four, it's all about creating these dynamic data networks for each, clouds for each individual patient to optimize wellness and minimize disease. It's really skeptical about how we currently are carrying out clinical trials. 30,000 patients, give them a drug, give them a placebo, extract the information in curves and make inferences, A, about the population, and B, about whether the drug is successful or not. Doomed to failure because every patient is unique genetically and environmentally, and you can't average them. So what P4 medicine does is takes 30,000 patients, assesses each of them individually, and aggregates according to shared properties that you'll be interested in with regard to drug response or whatever you wish. And of course, the patient-activated social networks are really key. So let me just say that the evolution of P4 medicine has been really fascinating. So we had formulated it in a conceptual way in the mid-2010s, 2005 or so. And of course, the really interesting question is, how would you go about implementation? And it was in 2008, an absolutely spectacular event happened where I met the Minister of Economy from Luxembourg, who was really interested in bringing modern biology and medicine to Luxembourg. And in return for that, he was willing to give us $100 million over a period of 5 years to focus on the development of both technologies and strategies for P4 medicine. And of course, these enabled us to generate 10 new systems-driven technologies and strategies that really put P4 medicine at a tipping point where it was really beginning to affect the practice of healthcare. And these are just a few of the things we looked at. We could, in animal models, look at disease dynamics and understand very early disease mechanisms. Enormously fruitful as long as the model organism diseases were orthologous to their human counterparts, and we can make them orthologous now. Number two, we kind of invented the technique of sequencing entire families because that gave us the ability to identify disease genes really rapidly. So for example, we've looked recently at 50 families with bipolar disease, and we've been able to show that each of the affecteds have multiple genes that are all related to neurotransmitters in really interesting ways, and it opens up whole new paths to thinking about what bipolar disease is all about. We can take a disease and stratify it into its different subtypes for impedance match against drugs. We can look at the disease-perturbed networks and make predictions about likely targets for as drug candidates. And of course, you know, Big Pharma today is very conservative in the candidates it employs. They come from one of 5 different protein classes for the most part. This is an entirely new way of approaching things. Longitudinal studies I'm going to talk about in just a few moments. But the one study I'll talk about that really is beginning to transform the healthcare system is in blood diagnostic. The problem was always if you compared normal bloods with diseased bloods, and you could look at proteins or nucleic acids, whatever you wanted, there were enormous numbers of differences, and 99% of them were noise. How did you get that 1% signal? We figured out how to do that, and in fact, the most recent thing we've done with a company called Integrated Diagnostics is to create a panel of 13 blood proteins that has the ability to unerringly distinguish benign lung nodules from their neoplastic counterparts. And this saves the American healthcare system $3.5 billion a year because you can avoid an enormous number of unnecessary surgeries for diagnosis. And what was interesting is those 13 proteins, 12 of them mapped into 3 of the major disease-perturbed networks in lung cancer, and that means we'll be able, 1, to do extremely early diagnosis, and 2, we'll be able to follow the progression of the disease and, in turn, its response to therapy as well. So if you think about P4 medicine, it really has 2 central essences. One is quantifying wellness, and the other is demystifying disease. And I think what's interesting to point out about contemporary medicine is it's 98% about demystifying disease. And in fact, all of the federal funds that go into healthcare go to the disease side of it. And in fact, on the other side, the wellness, it has a dubious reputation because of lots of quacks that are out there. So what we need to be able to do is quantify wellness. And I'm going to tell you exactly how we're going to go about doing that. But the really important point is I make the prediction that over the next 10 to 15 years, there will emerge a wellness industry that will, in that period of time, almost certainly far exceed the market cap of the disease industry, namely the current healthcare industry. And the wellness industry will do 2 really important things for healthcare. 1, it will let us optimize human potential, and I'll tell you exactly what I mean by that. And 2, it is going to transform how we look at disease. And it can do it in ways that those who focus on disease today could never do it. So I'll go on and explain that in some detail. But if there's any more compelling argument for the need for wellness, really effective wellness, it's this particular observation. If the trends of the last 10 years in extending life expectancy continue into the future, you can expect half of the children born today to live to be 100. And let's hope that they live to be 100 physically and mentally effective. So what we are proposing is we're going to set up a longitudinal study of 100,000 well patients that will last over an extended period of time. And I'm going to describe this and how I think it's going to absolutely transform the healthcare system. So the measurements we'll make are, 1, we'll determine a complete genome sequence for each individual because that's the framework against which you integrate all of the other data types. 2, we'll look at lab results from blood and urine and stool, and clinical chemistries, 1,200 metabolites, key organ-specific blood proteins that give us a sense for heart and liver and brain as well, to wellness to disease transitions. We're going to look at the gut microbiome. We'll carry out self-tracking and a number of other types of things. And of course, if you think about this proposal, you'll realize that in a relatively short period of time, if we can start today with 100,000, we'd be able to look back and say they fell into 2 different categories. Those individuals that remained well or improved their health, in those individuals who transitioned into disease. So with regard to those individuals who remain well over an extended period of time, we can use those data to create multi-parametric parameters that will allow us to diagnose wellness, and I'll argue we'll be able to deconvolute from that multi-parameter metric both the physiologic and the psychological aspects of what it is to be well, quantitating fundamentally both sides. Now, one of the reasons I think that's possible is we've recently created a really effective diagnosis for post-traumatic stress syndrome, separating Afghanistan soldiers that are normal from those that have PTSD. And PTSD is primarily a psychologically diagnosed disease, so and a lot of the markers have to do with stress and things like that. But the really important point is wellness is a much more sophisticated concept than just the absence of disease. And I would argue that each of us has a wellness well. It is of different depths for each of us. I would argue that most of us are near the bottom of this wellness well, and with the right parameters and behavior changes, we can elevate you to the very top of that well. And the depth of your well, I'll guarantee you, is genetically determined, and there are ways we can begin looking at that as well. Now, on the other side, if we go out and see that we're looking at individuals that have transitioned to disease, we can follow the every 3-month metric measurements back to the point of divergence of wellness to disease. We can begin to look at those early disease mechanisms and come to understand the diagnostics and the therapeutics that will be necessary to change that sick individual from a disease to a wellness, back to a wellness trajectory, and save the healthcare system all of those downstream dollars that would be required if the individual remained sick. So what we really plan to do then is to integrate a lot of these different data types together, and from either single data types or integrations of 2 or more data types, we can actually identify actionable possibilities. These are possibilities the individual can use, again, either to optimize wellness and or to minimize disease. We're using coaches with MD advisors to bring these actionable possibilities back to the individuals and try and persuade them to act on their actionable possibilities. And one of the most important things we see is with each 3-month tranche, you have the ability to look back and determine whether your changed behavior changes your chemistry. And it does in many, many examples. And there's nothing more inducive to behavior change than positive reinforcement like this that occurs in a repetitive type fashion. So we plan to start with about 100 individuals we have already. Next year we'll go to 1,000, the following year to 10,000, and later to the 100,000 individuals. And as I said, we've started already with 107 individuals in March. So we've gone through 2 separate data collections. And these are just a specific listing of various of the metrics that we've collected. And those are all things that I talked about earlier on. So we're using self-tracking, and we're using both personal history and psychologic metrics for analysis as well. So what was interesting from the very first tranche of data we got, namely the clinical chemistries, was that every single individual had multiple actionable possibilities. And this really emphasizes the fact that you may think you're well, but you have to ask yourself, how well are you really? And that's, again, why the metrics for wellness are important, to figure out where, for each of you, you are in that well and how much your human potential can be improved. So 90-some% had nutritional limitations, and 50 to 60% had cardiovascular, inflammatory, and prediabetes kinds of conditions. And let me talk about some of the actionable possibilities. We found 2 people with frank diabetes that had been totally missed by their physicians. I mean, it's crime. And boy, I'm not the coach, but I would have suggested changing physicians if they missed something like that. This is really a terrific yuppie example. So we had 3 individuals that had off-scale mercury levels. And I'll show you the results from one of them who ate tuna sushi 3 to 5 times a week. And he changed from tuna to salmon. And he was able, in 1-2 month period, to half the level of mercury. And the other 2 that were in a similar boat did similar kinds of things. So you can, and mercury is not good for neurons, that I can tell you. We found that a lot of people were deficient in vitamin D, and many of them were really severely deficient in vitamin D. And let me just say, being low in vitamin D has a lot of consequences that you don't want to be engaged with. But the really important point is you can't just say, oh, it's Seattle, you never see the sun, you don't have any vitamin. Because there are 2 components to it. There are 6 genetic variants in 3 genes that block vitamin D absorption. Okay? So that means it's a little more complicated than you just live in Seattle. And what's really striking is if you look at the individuals that have lots of these blocking variants, as compared with those that have 1 or a few, you can see in the blue line, the first levels of vitamin C were strikingly different in accordance with how many risk alleles you had. And you can see that we were successfully in pushing the levels up for both categories of people. But the really important point is, if you have a lot of those variants that block vitamin D absorption, you need to take megadoses of vitamin D to get yourself up to a normal level. If you have few, you take just ordinary doses. And in fact, you have to know which is which, because over an extended period of time, the megadoses can be toxic for people that don't need the vitamin D levels. And this is one example where we were able to push most of these 107 pioneers from the red bad side up into the green good side. Namely, they had levels that were appropriate. A second thing is we were able to use genetics and the Human Genome Project to do some really interesting things. And let me tell you, to a first approximation, there are 2 kinds of variants. There are a lot of common alleles that if you do very large population studies, some of which can be associated with disease, but they have very, very small effects, okay? Then there are another set of alleles that if you have 1 of them, it has an enormous effect, okay? And I'm going to give you an example of both of these cases. So we found 2 individuals that had hemochromatosis. So this is a genetic disease which facilitates the uptake of iron and moves people with 2 bad copies of this gene up to levels that are toxic most of the time, but not all the time. So again, you have to have the combination of genetics plus the clinical chemistries of how much iron you actually have. And again, what's insidious about hemochromatosis is it generally occurs in your late 40s, your early 50s. And it's insidious. It starts by destroying your pancreas, then your liver, and then goes to the heart. And usually it's your heart symptoms that expose it. But by the time you have the heart symptoms, you're a chronic invalid for the rest of your life and in serious trouble. So what we were able to demonstrate is, indeed, 2 individuals did have both bad copies of the HFE gene. But if you bled them initially once every 2 weeks for a period of months, you could bring it down almost to normal. What was interesting is 12 individuals had 1 copy of this variant. And you can see that their levels are intermediate, and we really raised the question of whether we should bring down those iron levels as well. So this is the most common Caucasian genetic disease. 1 individual in 10 has at least one bad allele here, and it is deadly if it's left untreated. The other key thing that all of these 14 individuals can do is have their families checked, because if you're a heterozygote, you want to know if your wife or your husband is also a heterozygote. You've got a 1 in 4 chance of having defective kids, if so. And the small variants that are very common are really interesting. And here's an example of a study that analyzed almost 200,000 individuals, half control and half the experimental. And the question was, what are genes that have an effect on the level of cholesterol? 72 loci were actually identified. And interestingly enough, what you can do with those loci is you can note first that they can either increase cholesterol levels or decrease it. You go either way. But you can use the probabilities of these things to add up for each individual a score of how likely you are to have trouble with cholesterol accumulation. And what's really striking, and in this plot, again, red is bad cholesterol and green is good, and the bad genes are to the left and the good genes are to the right. So in a gene like HDL, you want to have high levels of HDL. That's good for your cardiovascular disease. You can see in the first 12 or 13 people we looked at that there was more or less a linear plot, but 2 individuals, one was really off scale above it, overperforming what his genetics said. And it turned out he is a superb athlete, a triathlon winner, all sorts of things. So your genetics can overcome your genes. Really, now, you don't need to go quite to the triathlete category. But the other one is this underperformer over there. And what's interesting about that underperformer is about 4 years ago, he went through triple bypass surgery, which is a procedure for cleaning out cholesterol in key arteries and so forth. And what it almost certainly means is there are other genes we don't know about that can lead to these bad effects. But the other thing that one can do, because we have more than 2,000 complete normal human genomes, is we can, for those 2,000 genomes, add up for this across the 72 genes and score how each of that normal population is with regard to this trait. And then we can take the individuals that we're interested in and match them against that normal control. So if on the one hand you're way above average, almost certainly you're not going to be able to deal with your cholesterol by diet alone. You're almost certainly going to need statins or the equivalent. Whereas on the other side, you probably can handle it with diet alone. And what's really interesting is we have probably 50 diseases for which we have lots and lots of genetic markers. So in principle, we can tally the scores for each of the individuals and keep them for the individuals and report them once the diseases they have become actionable. For example, we're not going to give Alzheimer's reports now because there's nothing you can do about it. But in 5 years, if there's something, those people will be notified and they can be first in line to do things. Athletics is really interesting because there are more than 300 variants that control various kinds of athletic injuries. And what's interesting about all of these athletic injuries is if you know you have the gene defect, there are really good things you can do to assure you don't get ACL tears or whatever you might be worried about. And the other point that's really interesting is there are variants, for example, this one that increases significantly the production of a particular collagen that gives you a 10-fold less likelihood of an ACL rupture just by having more collagen. So I can see when the pro football coaches are going to be looking at genetics, deciding who really is going to get the short-term and long-term contracts. So the important point is that every single one of the 107 person tests, multiple actionable tests, and the other point is we're seeing the actionable possibilities change as their environments change. So you want to go on making these dynamical measurements. I'd say the 107 we have learned two really important lessons. I think they understand really clearly your genes aren't your destiny. They determine your potential. You can overcome many genetic limitations, certainly not all, but many. And the second thing is, almost to a person, they've realized that the key to their health is going to be taking individual responsibility. And I would say that's really the key to the healthcare system and turning around the ever-escalating costs of medicine as well. So I think there are three classes of opportunities. One, if you think about it, this 100K project I've discussed is a perfect pilot program for P4 medicine. And as it inserts itself into the healthcare system, I think the fundamental principles of P4 medicine will be introduced. That is, improving the quality of healthcare, decreasing the cost, and promoting innovation. I would say even more specifically from the healthcare point of view, one, we're going to get a catalog of all these actionable possibilities that will give us an enormous repertoire of information for dealing with people in the future. So right now, we have a graph database of 250,000 nodes and 4.5 million edges. And these are things that connect actionable possibilities with specific genetics and specific environmental possibilities. So we have the ability to screen an enormous number of actionable possibilities. Number two, we're going to be able to look at each individual and do this optimization of wellness, minimization of disease through encouraging them to act upon the actionable possibilities. Three, the idea that we can get this metric for measuring wellness and finding out where you are in your well and how effectively we can push you up to the top of that potential is really going to be important. I think being able to follow disease transitions, wellness to disease transitions, learn about the early mechanisms and how we can subvert the whole disease process very early in its process and all. And here's an area that is enormously important, and that is using old assays, but even more important, developing new assays. And ultimately, what we have to be able to do is convert essentially all the assays to a smartphone so people can go home and with a drop of blood can make all of the different kinds of measurements. And it won't quite be that easy, but I think this is absolutely a critical point of view. And finally, what about societal implications? Well, I would say there are a whole series of fascinating societal implications. One, if we succeed with the digitalization of the assays and so forth, we'll essentially be digitalizing medicine. And this, I think, will lead to a real democratization of healthcare across the world. The database will give us the possibility of innovation through using all of that information to create the wellness companies of the future or wellness to disease transition companies of the future and so forth. I argued that the wellness industry will exceed the disease industry in the future, and we're making the companies that'll be the Microsofts and Googles of the wellness industry as we speak today. It's an enormous opportunity. In competition, usually economic well-being comes from technological advances. And of course, the steam engine introduced the Industrial Revolution. It was a macro invention that spawned enormous micro inventions. I'll argue P4 medicine is a macro invention that's going to spawn enormous micro inventions for the wellness industry. Optimizing human capital. I mean, a really interesting point is people who don't function well at their jobs cost industry $1 trillion or more a year. And there are two ways you cannot function at your job. You can be absent, or you can be present but non-functional, and that's called presenteeism. 82% of the job loss falls in that latter category. And I think by improving human capital, we'll be able to really change those numbers strikingly. And of course, the financial crisis with the changes that will come from P4 medicine, and I think the new sense of responsibility individuals will have for their own health is really key. We're starting a wellness company now that is focused on consumers, and I think it's this company that is really going to be the key to the digitalization and the globalization of this new kind of healthcare. And I'm optimistic that in time it can lead to a democratization of healthcare for the developing as well as the developed countries, quite obviously. And I'll just close by saying I've never worked with a better group of people in taking an exciting project that 9 months ago was nowhere to the exciting kind of conclusion we have now. And I'll make the obvious, we're just beginning with the detailed integrative analyses. So the examples I've given you are just the front end, simplest kind of examples of opportunities we're going to see. So thank you very much for your attention. What a great question. So you must know the answer. So of the 107 individuals we recruited, most of them were my friends. And I would say 50% of them did it because of me, and they were quite skeptical and not particularly interested in doing this, but they agreed to do it. I would say virtually all of them have the traits I described here of taking on their own wellness and being convinced that this study has fundamentally changed their life. Now, the question you can ask that I still think is legitimate and we don't know the answer to is, look, all of those people are middle class, upper middle class. What about other classes? And I can't answer you that, but we will begin doing this next year, some big studies with clinical centers and disease populations that they've selected, so we'll have the chance to look at those kind of individuals as well. But I'll tell you what I think has really been the most transformational single factor is the coaching, and we hired superb coaches, and our ability to enunciate the actionable possibilities and the ability to see what you've done has changed your chemistry. So better living with chemistry, right? Yeah.
What about alternative medicines, like acupuncture, massage? Does that fit at all in your analyses?
Well, what we'd like to be able to do is quantify anything we think is legitimate wellness. So I'm pretty convinced acupuncture is really real, and we don't understand very much about it, and I think that is something we really could quantify. Some of the other things I think are on the fringe, and whether they're going to be legitimate or not. I mean, that's why, when I started this company, we're now having a big debate. There are a lot of people that want to throw out the word wellness because they think it has such a stigma attached to it. And myself, I think it's the wrong way to go. I think you make wellness a good word by quantifying it, but we'll see what happens. Yeah.
Hold on.
Oh, wherever.
Hi, over here. I think that there's an issue sometimes where we tend to oversimplify looking at certain metabolites in our body. So if we're low or high in something, simply applying one specific intervention should do the trick is like I said, a little bit of an oversimplification. And I want to point out a vitamin D example to highlight that, because as you know, vitamin D, the same precursors that are used to synthesize vitamin D are also used to synthesize cholesterol. So giving mega high doses of vitamin D may in fact correct vitamin D deficiency, but how do you know that that's really what you should be doing if that's also going to affect your blood lipid levels?
Sure. I think that's a great point. And what I find really exciting about this study is we can do a lot of N-of-1 experiments that can test those kind of hypotheses. So, for example, in those individuals that we've given meta doses of vitamin D to, we have looked at cholesterol levels and things like that and have seen no significant change in probably 10 or 12 individuals that we've looked at. But I do agree with you, nutrition is in the dark ages, and I think this is one way of beginning to cast light on things that will be correlated, things that we have to be careful of, and so forth. But the important thing is to make all the measurements so you can see how all the things are interconnected and what doing one thing does to another. I agree with that completely.
Hi, over here, on your left.
Oh.
Right here. Way left.
Way left.
So I have a question that I think is kind of a sociological question, and it has to do with moving towards a world where everybody gets their genome sequenced. And I think I feel like, if, based on kind of, the kind of thinking behind like the movie Gattaca, and do you think that there will be a societal response where people just kind of don't want to know what's in there, especially when you gave the example of pre-screening parents before they have kids? I mean, what kind of probabilities do you think people will find, because there are so many things that can go wrong, and do you think that people who really probably would be okay might decide not to have children, or do you think that maybe people will not want to know and resist the idea of getting that information because ignorance is bliss?
So that again is a really interesting question. So with 107 individuals, we've had 2 extended discussions on how much information do you want to know. And I would say there are about 20% that don't want to know anything bad. And that is really a fuzzy definition, I must say. We'll have to be very conservative with that. But I'd say 70 to 80% want to know everything so they can act upon it. And about a year and a half ago, we had a family retreat, and 40 members of my family came together. And we had this wonderful 3-hour discussion in the evening. I proposed doing our family genomes. And we talked about genetic discrimination. We talked about how it would change your behavior. We talked about genetic determinism. I mean, it was an incredibly interesting discussion. And at the end of it, all of the members chose to have their genomes done and the genomes of their kids done. But a number of them opted out of getting, again, the bad kind of results. So our feeling is, in a sense, each of the individuals owns their own data. And if they want it, we're more than willing to give access to it. But we have warned them that some of you may not want to know some of the things that are at data, and you really have to think it through. So we're in a complicated, fuzzy area. With regard to genetic discrimination, GINA, a law that prevents health insurance discrimination either from employers or insurance companies, and Obamacare has exactly the same provisions. But there are some aspects of healthcare, like long-term disability insurance, that aren't covered. So we really have to extend that and make sure those things are covered as well. So it's a complicated, ongoing question, and we just try and give people as much information as we can and let them make up their minds about what they'd like. What I will say is younger people have far less reservation about learning about themselves than older people do. It's really quite a striking. So the age range in our group is 21 to 93. So we covered quite a spectrum of people.
So you spoke a little bit about, I'm right here. You spoke a little bit about the possibility of discrimination, but can you talk a little bit more about the balance that we get on the good side with gathering all this information, using it all, but what the damage that potentially does in terms of individual privacy? So having all this information available creates more than just discrimination in terms of the possibilities for misuse, and what does the future hold for privacy if this is going to be the future?
Everything that all 107 agreed to have their data used in analyses after de-identification. And I think we have really good, effective ways of de-identification. And of course, the larger the population gets, the more meaningful de-identification is. So we're aware of that. But people have agreed that if we can de-identify, and if we have their data in a really secure place, and we hired a really good IT company to try and break into our database, and they failed. So at least that's one metric that we've done a good job on the security side of things. So these people, anyway, have agreed to have their data used. My own feeling is some people say the patient should own and control completely the data. I couldn't disagree more. I think they can have access to the data, but the data we have to be able to use so we can create a better medicine for your kids and your grandkids. And I think that's really important. And if you feel strongly about that, then you shouldn't participate in these kind of studies. And we've run into no one that fell in that category so far. But as we extend the studies, I'm sure we'll find people that will have that reservation, and their minds will be made up, and that's fine. I think that's your choice.
Can you, over here, can you give us a little bit more about the demographics of your 100? You're giving us sort of piecemeal info. And then a second question, just quickly, following on what you're just talking about, data being privatized or being global. Are you working with any insurance companies currently to look at how they might use this to reform how they decide to make policy in terms of healthcare?
So we have talked to a number of insurance companies. Most of them are very conservative and aren't interested in listening to new ideas like this. One exception has been Aetna, and I think actually they're a really interesting possibility. They have an innovation center that I think is doing some absolutely incredible things. So I'm going to go back there and talk about it. But I want to do exactly what you said. I'd like to work with them to write a new insurance policy that takes into account people undergoing these tests are really going to save them a lot of money. So give them a break on their insurance. But most insurance companies aren't ready to listen to these kind of things. And your first question was? Yeah, this first study was just a study to see operationally what we had to do. So I would say 70% of the people are from Seattle, and the remainder are from San Francisco and San Diego. 40, 60 men, women. We have probably 4 or 5 African Americans, a lot of Asians. So, but there was no attempt to get geographical diversity. As we broaden the studies out, and as we go to other countries, we'll start to bring in a lot of geographical diversity. We've had individuals from 15 countries contact me about wanting to carry out similar studies in those countries.
At the risk of driving anybody crazy, we do have another posting session.
Can I ask one question? One last question, potentially?
Where?
So I've been patiently waiting for about the last 4 questions. Over here.
Let's see.
Lee, over here.
Oh,
so,
okay.
So this technology, you've really laid out beautifully how it applies to chronic diseases. What about infectious diseases and starting to analyze things, for example, to take an example from right now, why certain people survive Ebola and others don't? Is that something that you've thought about in terms of expanding it to infectious diseases?
Absolutely. I think infectious diseases are a natural. And I remember talking with Tony Fauci 12 years ago. He's the head of the National Institute of Infectious Disease, arguing the best thing you could do for infectious disease is put a lot of money into a systems approach to understanding vaccines. They're just now getting around to doing that. So I would say a system study of infectious disease is in the dark ages, and it's something we're going to be partnering with an institute just across the block to begin taking these systems approaches to try and understand them. But I think it's really a crime if you think about HIV, if you think about malaria, if you think about TB, and more recently Ebola, it is absolutely a crime. We don't understand how to make the vaccines that can transform those. And we don't. We spent billions of dollars on drug companies doing what Jenner did back in 1796 with almost without alteration. And now we can change that. And it's really a unique and very important opportunity, particularly if you're going to be going to the developing world and everything. So I agree with you completely.
Lee, thank you very much.
You're welcome.